Language-to-language translators in DNA-RNA protein systems point to intelligent design. The intricate processes that sustain life are nothing short of miraculous in their complexity and precision. Among the most astonishing features of biological systems is the DNA-RNA-Protein mechanism, which operates as a sophisticated information processing system within every living cell. Central to this process are what can be metaphorically described as language-to-language translators, molecular machines that convert the genetic code in DNA into functional proteins. This translation process is so complex and precise that it strongly suggests the involvement of an intelligent designer. In this video, we will explore how these language-to-language translators in the DNA-RNA protein system point to intelligent design and why this interpretation is a compelling alternative to purely naturalistic explanations. The DNA-RNA protein system, an overview. The central dogma of molecular biology outlines the flow of genetic information within a biological system. DNA is transcribed into RNA, which is then translated into proteins. DNA, deoxyribonucleic acid, contains the genetic blueprint for the development and functioning of living organisms. The information encoded in the sequence of nucleotides in DNA is first transcribed into messenger RNA, mRNA, through a process called transcription. The mRNA then carries this genetic information to the ribosomes, the cellular machinery responsible for synthesizing proteins. In the ribosome, the sequence of nucleotides in mRNA is translated into a sequence of amino acids, which then fold into functional proteins. This process involves transfer RNA, tRNA, molecules, which act as adapters, matching specific amino acids to the corresponding codons, 3 nucleotide sequences, on the mRNA. This translation process is highly precise and complex, resembling a language-to-language translation system, where the language of nucleotides in DNA, RNA is converted into the language of amino acids in proteins. Language-to-language translation, a hallmark of intelligent design. 1. Complexity and specificity in the translation process. The DNA-RNA protein translation process is not just complex, it is also highly specific. Each codon in the mRNA corresponds to a specific amino acid, and this correspondence is maintained with incredible accuracy. The tRNA molecules are responsible for ensuring that the correct amino acids are brought to the ribosome according to the mRNA sequence, and the ribosome itself orchestrates the entire process with remarkable efficiency. This level of complexity and specificity is analogous to human-designed language translation systems, where meaning is precisely mapped from one language to another. In human experience, such intricate systems of translation are always the product of intelligent minds. Whether in computer code, linguistic translation, or other forms of information processing, complex translation systems require a designer who understands both the source and target languages and can create the necessary mechanisms for accurate conversion. 2. Irreducible complexity of the translation machinery. The molecular machinery involved in the translation process is a prime example of what biochemist Michael Behe terms irreducible complexity. Irreducible complexity refers to a system composed of several interacting parts, where the removal of any one part causes the entire system to cease functioning. In the case of the translation process, the ribosome, tRNA, mRNA, and various enzymes must all be present and working together for the process to succeed. If any one component is missing or malfunctioning, protein synthesis cannot occur. This irreducible complexity poses a significant challenge to naturalistic explanations, which typically rely on gradual, step-by-step evolutionary processes. The translation machinery could not have arisen through a series of small, incremental changes, as the system only functions when all components are in place. This suggests that the translation system was designed as a complete, functional unit from the start, pointing to the involvement of an intelligent designer. 3. The information content in DNA and its translation. The information content in DNA is another compelling reason to infer intelligent design. DNA is often described as the language of life, with sequences of nucleotides encoding the instructions for building and maintaining living organisms. However, this information is useless without a mechanism to translate it into functional proteins. The existence of the translation machinery implies that there is not only information in DNA but also an understanding of that information within the cell, an understanding that allows the cell to read and interpret the genetic code correctly. This interplay between information and translation is reminiscent of how languages work in human communication. A message written in one language requires a translator who understands both the source and target languages to convey the intended meaning accurately. The presence of such a sophisticated translation system in cells strongly suggests that the information in DNA was intentionally designed to be read and translated, rather than being the product of unguided processes. 4. The principle of sufficient reason. The principle of sufficient reason states that everything must have a reason or cause for its existence. When applied to the translation machinery in cells, this principle asks us to consider what could be a sufficient cause for such a complex and precise system. Naturalistic explanations, which rely on random mutations and natural selection, struggle to account for the origin of such an intricate translation system, especially given its irreducible complexity. Invoking intelligent design as the cause of the DNA-RNA-Protein translation system is not an appeal to ignorance but a reasoned conclusion based on the principles of causality and reason. It is a metaphysical argument that seeks to provide a sufficient cause for the existence of life's most intricate information systems, consistent with the evidence of purpose and design observed in nature. Conclusion. The language-to-language translation system in DNA-RNA-protein mechanisms provides one of the most compelling pieces of evidence for intelligent design. The complexity, specificity, and irreducible nature of the translation process suggest that it could not have arisen through unguided natural processes. Instead, it points to the involvement of an intelligent designer who created the translation machinery as a complete, functional system from the start. This argument is not a God of the gaps argument but a reasoned conclusion based on positive evidence. It complements, rather than contradicts, scientific inquiry by offering a coherent explanation for the origins of life's most intricate information systems. In the context of everything we know about complex, functional systems and language translation, the inference to intelligent design stands as a robust and compelling explanation for the existence of the DNA-RNA-protein translation system. Language-to-language translators in DNA-RNA-protein systems point to intelligent design. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts. If you would like me to respond to your comment, please make a level 1 comment directly below the video. I will respond to all polite level 1 comments even if it takes me a while, due to other commitments.